Morning, my dear students. Welcome to our zoology online class. In the last class, we studied about some important lymphoid organs. Okay, so just remember that all the points. See, the lymphoid organs are classified into two major categories: primary lymphoid organs or central lymphoid organs, and the second one is secondary lymphoid organs, otherwise called peripheral lymphoid organs. Okay, and in the last class we studied two important secondary lymphoid organs. Okay, two important secondary lymphoid organs and two important primary lymphoid organs. Understand? See, the lymph node and spleen, these two are the important secondary lymphoid organs. Understand? The last class we studied. Then thymus and bone marrow, these two are the important primary lymphoid organs. Understand of you? So in the last class we studied about the structure of all these lymphoid organs. Okay, and we studied some important lymphoid tissues also. Okay, in the last class we studied about some important lymphoid tissues also. Example, the Peyer's patches, tonsils, malt, galt, belt. These are some of the lymphoid tissues. Okay, my students. Okay, today we are going to study about two more topics. The first one is lymphoid cells. Cells involved in our lymphoid system or immune system is called lymphoid cells. Understand a few? So today we are going to study about the lymphoid. First we are going to study about lymphoid cells. See, you all know there are three types of red cells are present in our body: WBC, RBC and platelets. Okay, RBC, WBC and platelets. There are five types of WBCs are there. Okay, lymphocyte, monocyte, basophil, eosinophil and neutrophil. These are the five types of WBC. So all the five types of WBCs and RBC and the platelets. So all these cells are involved in our immune system. So first we are going to study about the structure of lymphocyte. Okay. See, 20 to 30% of the WBC molecule is lymphocyte. Okay, so lymphocyte, monocyte, basophil, neutrophil, eosinophil. These are the five types of WBC. Here, 20 to 30% of the WBC molecules are this lymphocyte. Understand? So the first point is the lymphocyte consists of a large nucleus. A large nucleus is present in the center region of the lymphocyte. Understand? And the nucleus is surrounded by cytoplasm. The amount of cytoplasm is very little. So the size of the nucleus is very big. Size of the cytoplasm is very small. Understand? So this is the first point about this lymphocytes. Understand? Then there are two types of lymphocytes are present in our body. One is B lymphocyte and the second one is T lymphocyte. B lymphocyte and T lymphocyte. Here both B lymphocytes and T lymphocytes are produced inside the bone marrow. You all know bone marrow is one of the examples for the primary lymphoid organ. Okay, so the both lymphocytes are produced inside the bone marrow, but the B cells only mature inside the bone marrow. So the production and maturation of this B lymphocyte takes place inside the bone marrow. But the production of T lymphocyte takes place in the bone marrow, but their maturation takes place inside the thymus. Okay, so thymus is also one of the example for the primary lymphoid organ. So I hope you are understood this point. So there are two types of lymphocytes are present: B lymphocyte and T lymphocyte. Here the production and maturation of B lymphocyte takes place inside the bone marrow, but the production of T lymphocyte takes place inside the bone marrow, but their maturation is inside the thymus. Understand up to? So I hope you are understood all this part. Then next one is, see, when a pathogenic microorganism or when an antigen is contact with our B lymphocyte, okay, with our B lymphocyte, immediately the B lymphocyte is activated. Because the outer surface of the B lymphocyte, a protein is there. This protein is called receptor protein. So listen to here. See, this is the B lymphocyte. Just imagine it's the B lymphocyte. So outer surface of this B lymphocyte, your protein molecule is there. This is called a receptor protein. Okay. So when this receptor protein coming to contact with an antigen or a foreign particles, immediately this B lymphocyte is activated. The B lymphocyte is activated. So this activated B lymphocyte immediately produce plasma cells. Produce one more cell is called plasma cells. These plasma cells produce more amount of antibodies. Immediately the plasma cells producing more amount of antibody. Understood you? So I will repeat this point. When a B lymphocyte, okay, is contact with the antigen, the receptor protein present on the outer surface of the B lymphocyte is making a contact, and immediately the B lymphocyte is activated. The activated B lymphocyte is producing plasma cells, and plasma cells producing more amount of antibodies. So these antibodies getting ready to fight with the antigen. Students, then one more important point. Some B lymphocytes will not produce any antibody. They do not produce any antibody, and they become memory cells. Okay, so the B lymphocytes which are not producing antibodies become memory cells. Understand a few? Then the T lymphocyte, the second type of lymphocyte is called T lymphocyte. Already I told you B lymphocyte and T lymphocyte. Then the T lymphocyte do not produce any antibody. It will not produce any antibody under any situation. And there are these T lymphocytes are in two forms inside our body. These T lymphocytes are in two forms. One form name is called T killer cells, and the second one is called T helper cell. T killer cell and T helper cell. Here this T killer cell is killing all the damaged or infected cells present in our body, destroying all the damaged or the infected cells of our body. Then this T helper cell is releasing a chemical-like substance. Name of this chemical-like substance is called cytokines. So this cytokines helps the B cell to activate. Helps the T cell to activate. So say repeat this last point. The T lymphocyte do not produce any antibody under any situation. And these T lymphocytes are in two forms. One is T helper cell and T killer cell. Here T helper cell are releasing a chemical-like substance is called cytokines, and these cytokines helps the B cell to activate, and the T killer cell destroying all the damaged or the infected cells of our body. Understand, my dear students? So that's all about the lymphocyte. So now we have completed one important cells of our immune system, the lymphocyte. Okay, many 1 mark questions are there, and this one of the very important 5 mark question. Understand, my dear students? Okay. Now we are going to study about some 2 mark questions. Very important 2 mark question. The first one is antigen. First one is antigen. See, the antigen means the substance which are reacting with the antibody or the product of our immune system is called antigen. The substance which are react with the antibody is called antigen. Okay. Then immunogen. Second one is immunogen. Both the antigens only, otherwise called immunogen. Okay. But the term, the definition is different. Antigen is only, otherwise called immunogen. But the definition is different. Here immunogen means the substance which can produce immune response is called immunogen. That means when this antigen enter inside the body, immediately our body producing the immune response. Okay, so for the production of immune response, one substance is needed. That substance is called what? Immunogen. Is called immunogen. So now we completed 2 mark question, antigen and immunogen. Then the third 2 mark question is heptons. Heptons means a non-immunogenic antigen. Okay, non-immunogenic substance, but it can react with the product of our immune system. But it can react with the product of our immune cells of our immune system. It is called heptons. Understand you? Then next word is called epitope. Listen very carefully. Epitope is the active part of an antigen, and it is an antigenic determinant. So listen. See, this is just imagine this is an antigen. So the outer surface of the antigen, one special part is there. These parts are called antigenic determinants. These parts are called antigenic determinants. Understand? So the outer surface of the antigen only, the antigenic determinants are there. So these antigenic determinants are otherwise called epitope. Antigenic determinants are otherwise called epitope. So epitope means the active part of an antigen. Then next one is paratope. Paratope is the active part of an antibody which making a contact with the epitope. So just imagine this is an antibody. This is an antibody. So this region is called paraplex region because this paraplex region only making a contact with this antigen in this epitope region. So this region is called paraplex. So paraplex means the active part of antibody making a contact with the antigen or epitope or antigenic determinants. Understand my dear students? So these are some important 2-mark questions: antigen, immunogen, reference, epitope, paratope. This epitope, paratope, sometimes it can be asked for differential question. Then next one is types of antigen. Antigens are classified into 2 important types. One is exogenous antigen, then second one is endogenous antigen. Here exogenous antigen means what are the antigens entering inside the body from outside is called exogenous antigen. Example for this exogenous antigen: microorganisms, pollens, pollutants, some drugs, chemicals. These are the example for the exogenous antigen. So antigen which are entering inside the body from outside is called exogenous antigen. Then next one is endogenous antigen. The antibodies which are produced inside our body, the antigen which are produced inside our body is called endogenous antigen. Example, blood group antigen. Okay, blood group antigen is the example for the endogenous antigen. Understand my dear students? So this is a 2-mark question. Types of antigen. There are 2 types of antigens are there: exogenous and endogenous antigen. Okay, exogenous means antigen entering inside the body from outside. Endogenous antigen means antigens formed inside the body. Is called endogenous antigen. Understand my dear students? Then next we are going to study about one very important 5-mark question: structure of antibody. Okay, and one diagram also there in your book. It is very important for 2-mark question. Listen very carefully. Antibodies are otherwise called immunoglobulin. Here I told you antigens are otherwise called immunogen. Like that, antibodies are otherwise called immunoglobulin. And these antibody molecules are produced by our immune system. Sorry, B lymphocytes. B lymphocytes of our immune system. So the B lymphocytes only produce responsible for the production of these antibodies. Understand a few? So whenever microorganism or the foreign particles or the antigens entering inside the body, immediately the B lymphocyte produce different types of protein molecules. These protein molecules are called antibody. These protein molecules will be called antibody. So whenever pathogenic microorganism enter inside the body, these B lymphocytes releasing this protein molecule. These protein molecules are called antibodies. Understand a few? Understand? Then, so antibodies are produced on exposure to antigen. When antigen is produced, enter, immediately antibodies are produced. Okay? Then, this antibody molecule, the structure of the antibody molecule was first explained by 2 scientists. Their name is Edelman and Porter. Edelman and Porter. So these 2 people really first explained the structure of antibody. Understand? Then, based on the structure and function, physiological properties, based on the physiological properties and biochemical properties, the antibody molecules are classified into 5 types. Antibody molecules are classified into 5 types: IgA, IgG, IgM, IgD and IgE. So these are the 5 types of antibody molecules. Understand my dear students? I hope you all understood all these points. Now, all clear? See. Actually, antibody molecule, the shape of the antibody molecule is Y-shape. Antibodies are Y-shaped molecule. And it is made up of 4 polypeptide chain. Each and every antibody molecules are made up of 4 polypeptide chain. 2 chains are very small chains, so they are called light chain. And 2 polypeptide chains are very big chains, so they are called heavy chains. So there are 2 light chain and 2 heavy chains are present in a single antibody molecule. So this is 1 light chain, this is second light chain. This is the 1 heavy chain and this is the second heavy chain. Understand a few? Then the molecular weight of this light chain is 25,000 dalton. The molecular weight of the light chain is 25,000 dalton. And the light chain is made up of 214 amino acid molecules. 214 amino acid molecules are present in the light chain. Then the molecular weight of this heavy chain is 50,000 dalton. 50,000 dalton, and this heavy chain is made up of 450 amino acids. 450 amino acids. Understand my dear students? So I hope you all understood all these points. Then disulfide bond. this bond is playing a very important role in the structure of antibody, because the light chain, one light chain is attached with one heavy chain by a single disulfide bond. Here also this light chain is attached with this heavy chain by a disulfide bond. Listen of you. Then both heavy chains are also connected by a disulfide bond. So the connection between one light chain and one heavy chain, and the connection between two heavy chains are by this disulfide bond. Understand, my dear students? Okay. Then next point. Each and every antibody molecule has two ends. One end is called N-terminal end. This end is called N-terminal end, and this end is called C-terminal end. N-terminal end and C-terminal end. Okay. Then single antibody molecules has two important regions. Okay. So one region is called variable region. So this is the variable region. This is also variable region, and this is the constant region. Okay. So the top region is called variable region, and the bottom region is called constant region. Understand? So in variable regions, some variations are there in the amino acid molecule. They told you it is fully made up of amino acids. Okay, fully made up of amino acids. So some variations or differences present in some region of the antibody molecule. So that region is called variable region. And in this region, there is no variations or there is no differences in the amino acids arrangement. So this region is called constant region. So this is the variable region, and this region is called constant region. This region is called constant region. Understand, my dear students? Then listen to the next point very carefully. See, one variable region of the light chains, variable region of the both light chains, and variable region of the both heavy chains, variable region of the both heavy chains togetherly forming an antigen binding site, So the variable region of both light chain and variable region of both heavy chains togetherly forming an antigen binding site. In that antigen binding site only the antigens are fitted. Okay, here I told you. So this is the antigen determinants. Okay, so in this region only the antigen determinants are fitted. The antigen and antibody making a contact in this region. Okay, so this is the region. Understand, students? So I hope you all understood the structure of antibody. Diagram is there. It is very important for 2 mark question. Understand? Okay, dear students. Next we are going to study about 2-3 mark questions. So the first one is binding force of antigen antibody reaction. So here antibody means the protein molecule produced inside our body. Antigen is the foreign molecules that are entering inside the body from outside is called antigen. Okay, so the antibody which are produced inside our body should destroy or eliminate the antigens. Okay, so in between the antigen and antibody some reactions are takes place. Finally, the antibody destroying the antigens. Okay, so the force of antigen antibody reaction is determined by 3 factors. Okay, so just imagine this is antibody, this is antigen. So this antigen and antibodies are making a contact. Finally, this antibody eliminating and destroying this antigen. So the force of reaction between this antigen and antibody is determined by 3 factors. The first factor name is called closeness. That means the antigen and antibody should fix very closely. Okay, closely tight. Then only the reaction becomes very strong. Okay, if any gap is there, the reaction is very slow. So the closeness is the first reason or first factor determining the force of reaction between antigen and antibody. The second one is non-covalent bond. Okay, so non-covalent bond is formed between the antibody and antigen. Okay, so the bond should be non-covalent bond. Example for non-covalent bond, hydrogen bond, electrostatic bonds. These are the examples for the non-covalent bond. Then the last one is affinity of antibody. Affinity means strength of antibody, power of antibody. So these 3 factors will be determining the force of reaction between the antigen and antibody. Understand, my dear students? So this is one of the very important 3-mark question, and it's very simple, easy question. So there is a reaction is present between the antigen and antibody. So the force of this reaction is determined by these 3 factors: closeness, non-covalent bond, and affinity of antibody. Then next one is types of antigen-antibody reaction. Next 3-mark question: types of reactions. Okay, see, in between the antigen and antibody there are 4 types of reactions are there. 4 types of reactions are there. The first reaction is called precipitating reaction. First reaction is called a precipitating reaction. Listen very carefully. When a soluble antigen is reacting with antibody, immediately one precipitate is formed. Immediately a precipitate is formed. So this reaction is called precipitating reaction. This reaction is called a precipitating reaction. And antibody which are bringing this precipitating formation is called precipitating. Listen very carefully. So the first reaction is called precipitating reaction. Here, when a soluble antigen is making a contact with the antibody, immediately a precipitate is formed. So this reaction is called a precipitating reaction. The antibody which are involved in this precipitating reaction is called precipitating. Then second reaction is called agglutination reaction. Second reaction is called agglutination reaction. Here agglutination reaction means when a particulate antigen reacts with the antibody, immediately a agglutination is formed. A particulate antigen reacts with the antibody, immediately agglutination is formed. Okay, so this reaction is called agglutination reaction. So this reaction is called agglutination reaction, and the antibody which brings this agglutination reaction is called agglutinin. Is called agglutinin. Understand? The third reaction is called opsonization. Here one substance is formed called opsin. Opsinin. When substance is produced, it is called opsin. So this opsinin enhance the antigen or ingest the antigen. Okay, or the antigens are coated by this opsin. Antigens are covered with this opsin. Finally, the antigens are destroyed. So this reaction is called opsonization reaction. And the last reaction is called neutralization reaction. Neutralization reaction. So in this reaction, the harmful effect of microorganism or the harmful effect of the antigen is eliminated or reduced or neutralized by the antibody. So this reaction is called neutralization reaction. Understand all of you? So the harmful effect of the microorganism or antigen is eliminated or neutralized or reduced by the antibody. So this reaction is called neutralization reaction, and now this antibody is called antitoxin. Antibody is called antitoxin. Understand, my dear students? So now we completed one important 3-mark question: types of antigen-antibody reaction. Okay, so today we have completed 2 important 5-mark questions. One is the structure of lymphocyte and the structure of antibody. These 2 are the important 2-mark question answers, and these 2 are the important 5-mark questions, and some 2-mark questions also studied: antigen, immunogen, vector, epitope, paratope. And we have studied 3-mark questions. The first one is types of antigen, second one is binding force of antigen-antibody reaction, and the types of antigen-antibody reaction. Understand, my dear students? So today itself try to complete all these questions. All the questions are very easy and simple questions. So study well. We will meet in the next class. Thank you.